301 and you're hitting a possum. And a poor little sucker crawls to the side of the road and keels over dead. And he's out there in the hot sun all day. And his immune system is not working because he's dead. So all the bacteria in his gut are now going to be able to replicate because it's warm, like putting them in an incubator, speeds up their metabolism, they replicate faster, and no immune system to keep them in balance. That's what starts the decomposition process, your own gut flora. That's why they can make Roman garum sauce, the fermented fish sauce. They take the fish, they throw them in a bucket, their own gut flora starts breaking them down. And if it's a sealed bucket, they'll ferment them into fish sauce. They put salt in there to steer the fermentation. We want the salt-loving bacteria to grow and do the fermentation. We don't want molds and other crap growing. So, anyways, so here's your roadkill out there. You go over and you poke him with a stick. He smells terrible and all this gas comes out. How they swell up? Because when the gut bacteria start digesting and fermenting inside of it, they give off gas, carbon dioxide gas. And also the breakdown products. So he smells from the breakdown products. So there's another test we used to do instead of API, it was called Intro-Tube 2. They don't make it much anymore. But I love the way they describe it. They have a little compartment with lysine in it. Lysine's an amino acid. And you test the bacteria's ability to digest lysine. If the bacteria digest lysine, it's because they have a gene for an enzyme. The enzyme is lysine decarboxylase. So what's it doing? It's chopping carbons off of lysine, right? If the bacteria decarboxylate lysine, it results in the formation of the end product, cadaverine. Sounds lovely, doesn't it? Cadaverine. That's why cadavers smell. Another one, ornithine. If the bacteria have ornithine decarboxylase and break down ornithine, it results in the alkaline end product, putrescine. Two lovely fragrances. And that's why roadkill and dead bodies stink if you don't get to them right away. So that's what your Enterobacteriaceae do. All right, so how can we differentiate E. coli from Serratia, from Salmonella, from Klebsiella, from Proteus, from whoever else? Look at the different sugars they can metabolize. Look at the different amino acids they can metabolize. They all can metabolize glucose. That's no help. They're all catalase positive. That's no help. But if you look at the sugars like lactose, and if you look at amino acids like lysine and ornithine, you'll see differences there. I actually have a pretty picture I could show you, but we'll just keep reviewing. Alrighty. Maybe I'll show it to you tomorrow. What else we got here? All right. Don't forget about your plates, your special media. What happens on your nutrient agar? Everything that we work with in lab should grow on nutrient agar. Salmonella grew, Escherichia coli grew, Staph epi grew, not as well, but it grew. And our unknowns also grew, right? That was easy. Those were controls. Then we have controls of the special media plates, our tri plates. And we saw if you grow E. coli on EMB agar, it comes out shiny green. Tells you it's E. coli. If you grow E. coli on MacConkey agar and on SS agar, it's going to make that milky pink color. Strong color reaction because it's a strong lactose fermenter. Salmonella. Salmonella grew on the EMB agar because it's selected for gram-negative enterics, but it didn't get a color change. Same thing on the MacConkey agar. It grew, but it didn't make a color change. That's because it's not a lactose fermenter. On the SS agar, it turned black from a different reason, not because of lactose, because there's hydrogen sulfide precipitate that it makes from the agar. It turns black. So we knew it was salmonella. E. coli didn't turn black. Serratia didn't turn black, or whatever it was. SFE didn't even grow. So some of the agars are selective. What does that mean? Promotes the growth of one, inhibits maybe the growth of another. You don't get a color change though. It's the differential agars that give you a color change. They're selective to start with. They'll select for gram-negatives or gram-positive staphylococci or whatever. But when you do have a particular organism growing, you'll get a color change. That's what makes it differential, a color change. What happened on the mannitol salt agar? It grows only staphylococcus because of the salt. Staph likes salt. If your staph are growing and the agar has no color change, they're non-pathogenic. But if they change the agar yellow, that means they're pathogenic because they're taking up the mannitol sugar in the agar and fermenting it, and that's what pathogenic staph does, ferments mannitol. So in that case, if you're gram-positive staphylococcus, mannitol fermentation means you're unfriendly, means you're pathogenic. Alrighty. Don't forget about some of the chicken bacteria. Salmonella comes from chicken. It's in this family. Campylobacter is food poisoning that also comes from chicken. Also happens to be gram-negative, but it's not in this family. What family is it in? It's in the same family as your spiral-shaped bacteria, I think, with Helicobacter. Is it? Or is it in the aerobic rods?
It's been in your Vibrios, if I remember right.
Yeah.
Campylobacter.
Just remember, it's not with Enterobacteriaceae. Although it's enteric bacteria for chickens, it's not in family Enterobacteriaceae. So you can see where classification might have changed over the years, too. Which
The
section is it in?
Helicobacter? Yeah, it's
Campylobacter.
oh yeah, it's in the gram-negative, gram-negative bacteria too, yeah. It's under spiral.
Not spirochetes, spirules.
Silicone?
Yeah, needleful spirules. Yeah,
Okay.
I was right the first time.
Yeah.
All you want to remember, Campylobacter comes from chickens, but it's not family Enterobacteriaceae. The one that's family Enterobacteriaceae that comes from chickens is who? Salmonella. Alrighty.
It's the Helicobacter that was Vibrio shaped.
Yeah, Helicobacter is also in the spiral
Yeah,
thing.
but it's a Vibrio shape,
Yeah.
like a curve, huh?
Yeah, it's more curved than spiraling. Okey dokey. We did that one? Okay, what does API 20E stand for? Anaerobic Profile Index, 20 tests for Enterobacteriaceae. What are you gonna end up with after you score your card? A 7-digit code. Based on what nutrients it digested and which nutrients it did not digest. Then we're gonna put it into the computer, and the computer is gonna tell us if it matches the profiles of other organisms of this species, or if it matches the profile of other organisms of that species. So it's gonna match them up. Okay, thing we did on the board today, resident microbiota, indigenous microbiota, normal flora, indigenous flora, commensal flora. Those are all terms for normal flora. What's the one that comes and goes? Transient. Okay, I think we covered it. So that shouldn't be hard. And there's only like one question about catalase, so don't even worry about it. Just remember which ones are your Enterobacteriaceae, and that Enterobacteriaceae are all catalase positive.